Hi everyone, we're into chapter 9, which is controlling microbial growth in the environment. Chapter 6 was all about how to grow microbes, especially in the laboratory. But now we're concerning ourselves with controlling their growth when we don't want them around. Table 9.1 is full of terminology that is important. So I need to go into that particular table and make sure that you're familiar with the different terms. I want to point out just a couple of things to you from this table. The first is the term side or cytal. This means that we're killing the microbes. For example, if you say something like bactericide, we're killing bacteria. A viricide is something that could kill viruses. Now, stasis or static at the end of a term is indicating that inhibits the multiplication, but it doesn't kill the organism. You might say bacteriostatic or fungistatic or viristatic, meaning the microbe won't multiply in those conditions, but you don't necessarily kill them. The other thing that it's important to point out is the difference between something like sanitation and disinfection. Those are pretty similar terms. Disinfecting means you're trying to get rid of most of the microorganisms, including viruses, on some sort of non-living thing, a desk, a counter, something in a doctor's or a dentist's office. You disinfect these particular objects and surfaces. Sanitation is pretty similar. We're saying that we're removing the pathogens from an object, but we're really referring to conditions that are specific to areas where the public has some interaction. There's a standard that's in place. Kitchens that are commercial kitchens must sanitize particular objects. Public restrooms, those are sanitized, but you would normally say you disinfect your own bathroom at home. Make sure you take a look at these terms and are familiar with what they mean. This next figure is pretty important. It's showing you what's called a decimal reduction time, or D value for short. How can you determine a D value from looking at a graph like this? Well, the first thing that you have to know is that decimal reduction time is indicating how long it takes to get rid of 90% of a population in the conditions given. When you get rid of 90% of a population, you decrease the population by 1 log value. For example, you go from 10 to the 7th organisms per mil in a particular solution to 10 to the 6th per mil. You've killed 90% of the original, only 10% are left. That million cells per mil is still a lot, but it's only 10% of what was 10 to the 7th. This 90% is killed in a given time, which is the D value. You need to look at the graph and to determine where has 1 log of organisms been killed. If you look at this particular graph, they've indicated it for you. The 10 to the 7th is present right there at the 2-minute mark. And the 10 to the 6th population is here at the 3-minute mark. That means it took 1 minute at this particular temperature to get rid of 90% or 1 log of the organism. Therefore, the D value on this particular graph is 1 minute. Under these particular conditions shown on this particular graph, we're able to reduce the population of organisms by 90% or 1 log in 1 minute. Make sure that you can look at a graph and determine the D value. Next, we're going to go through quite a number of antimicrobial agents in this chapter. What I recommend in order to organize your thoughts so you can answer future questions is to make yourself a table that lists the antimicrobial agent, whether it's physical or chemical agent. And in addition, you want to note the main mode of action, meaning what effect the agent has on microbial components. How are we killing the organism with this particular agent? Some of the major targets for microbes are going to be the cell wall, because that's how cells with them deal with osmotic stress. If you can somehow compromise the cell wall, you're going to cause the cell to become very sensitive to osmotic changes. The images that are shown in the bottom corner of these slides are cells that are breaking apart. They're micrographs of cells undergoing osmotic stress and essentially exploding. The cytoplasmic membrane is the semi-permeable barrier that regulates what's going into and out of the cell. So if that's damaged, homeostasis cannot be maintained, which will kill the cell. The viral envelope is similar. It's responsible for attachment because it has the ligands on the surface that'll become very obvious in chapter 13. The viral envelope is key. If you get rid of the envelope on viruses that normally have them, then the virus can no longer infect a cell. The other target might be proteins and nucleic acids, the large biomolecules of the cell. If you disrupt them, then of course the cell can't function properly and the cell is going to die. Some of the physical agents that can damage large cellular components, including the large biomolecules, are radiation and heat. There's also various chemicals that can interfere with these major molecules. If you alter the DNA, that can actually induce fatal mutations that kill the cell. You can also halt protein synthesis by damaging DNA. You can damage proteins themselves or the component of ribosomes. Ribosomes are composed of both protein and RNA. Different antimicrobial agents target large cellular structures and biomolecules. These are general cellular targets that cause very wide-ranging lethal problems for the cell when they're disrupted. When you're trying to select which agent is going to be the best for your particular application, and there's lots of different applications, you have to consider 4 major things. One is ideally the agent is not expensive. You want it to be pretty inexpensive so that it can be purchased in large amounts for your application. You want it to work fast. It's not helpful if you have to leave your agent on for hours or overnight in order for the activity to take place. You also want your agent to be stable during storage. For example, if you're a business and you want to purchase some of this agent, you don't want to have to keep purchasing it every week. You want to be able to purchase a supply and have it last in storage for a reasonable amount of time. Maybe that's a month, maybe it's longer, but you don't want to have new shipments coming in all the time. You also want it to be capable of controlling the microbial growth of concern. And finally, you don't want it to be harmful to people, animals, or the objects that you need to apply it to. Importantly, none of the antimicrobial agents that we'll consider have all of these particular components. You have to weigh the pros and the cons to pick the best one for your application. What else do you need to consider? You need to analyze what or where you're treating with the antimicrobial process or agent. What are you trying to disinfect or treat to control microbes? If it's a harsh chemical, or if you're considering extreme heat, you can't use that on humans or animals or fragile objects. You're going to have to choose something else. Then you need to consider the situation. If you're doing some sort of medical procedure, what is it? What kind of control is going to be important? A surgery is going to require procedures to get rid of nearly all the microbes in that surgical environment. The surgery suite, the skin where incisions will be made, hands, equipment, all need antimicrobial treatments to prevent subsequent patient infections. Different treatments are appropriate for the different objects and people. For another minor procedure, sterility is not perhaps nearly as important. Consider a routine blood draw. This is not conducted in a sterile environment. A clean and disinfected one, yes, probably, but not to the same degree as a surgery suite. You also need to consider what organisms are posing the most risk for your particular application. Microbes are susceptible to different degrees to the different processes and products. In general, we'll classify things as high, intermediate, or low efficacy. Those are going to be considered when we need to use these in a particular application. If something's a high-level antimicrobial agent, it pretty much kills all the pathogens, including any type of endospore. For example, these might be required for a lot of medical applications, but it might bring with it other problems. The agent might have some toxicity to humans. Maybe it's very unstable. You can't use a high-level antimicrobial agent under every condition. The intermediate level antimicrobial agents will kill the fungal spores and the protozoal cysts, along with the viruses and the pathogenic bacteria. These come into play quite frequently if you don't need something to be sterile. The low-level antimicrobial agents will kill the vegetative bacteria, meaning the metabolizing forms of bacteria, but not necessarily the endospores. They can kill fungi and protozoa and some of the viruses, but maybe not everything. Again, we have to consider, where are we applying this, and what do we need to do in order to be able to pick the correct agent to use for our application. This figure is from your book. It provides an indication of general resistance going from the most resistant, which is a prion, something we'll talk about later. A prion is an infectious protein, think mad cow disease. The resistance is indicated all the way down to the most susceptible microbes, which would be enveloped viruses, which are actually pretty fragile. So prions are the most resistant, then endospores come next, mycobacteria, the agents of tuberculosis and leprosy, cysts of protozoa, then the trophozoites of protozoa, think the Giardia organism that you drew during lab, then gram-negative bacteria, fungi, non-enveloped viruses, things like a cold virus, gram-positive bacteria, and envelope viruses. This figure shows the most resistant to the most susceptible. Depending on what microbe you are trying to kill, different agents will be appropriate. There are environmental conditions that affect how well an antimicrobial agent can work. Temperature and pH are important because they will affect microbial death rates. In general, as you raise the temperature, agents become more effective, but you may reach a point of no return. If you get it too hot, you inactivate the agent itself. For pH, there may be a range where the agent is useful, and then outside that range, it becomes not useful. Organic materials can interfere with the activity of some agents. Some agents are sensitive to the presence of organic materials. Think of blood, or the fluids that come from a wound, or food residue. These are filled with organic material that might interfere. The presence of these may be a problem. For some agents, they may not be effective treatment if there's a lot of organic material, so you have to consider that possibility. This particular graph is demonstrating the effective temperature on an antimicrobial agent on a particular strain of bacteria. We are looking at a particular strain of bacteria that has been inoculated to a level of somewhere between 10 to the 4th and 10 to the 5th per mil. After it was exposed to a particular agent, and that population of organisms was tracked over time, you can see that the organisms are killed faster at 45 degrees Celsius compared to 20 degrees Celsius. 20 Celsius is close to room temperature. Warmer temperatures in general tend to be more effective than cooler temperatures, as demonstrated here. This next concept is the biosafety levels. We've actually talked a bit about them in the laboratory already. There are 4 biosafety levels. Biosafety level 1, BSL1, is the lowest threat. These are organisms that don't typically cause disease in humans. You can imagine something like bread yeast, the cultures in yogurt, the normal E. coli that resides in our GI tract. Those would be BSL1. They are not going to cause a problem in a healthy human. BSL2 may be hazardous under specific conditions. These are microbes that could cause a problem. Typically, in a teaching lab, we use BSL2 strains that are attenuated compared to the wild-type strains in nature, but they still are of concern and must be handled appropriately to avoid risky situations. Think about bacteria like Salmonella or Shigella, or the organism that causes strep throat, Streptococcus pyogenes. These are considered BSL2. BSL3 are very high-risk organisms. They cause serious infections that are difficult to treat and need only to be handled in biosafety cabinets with trained individuals. BSL-4 are especially dangerous organisms that tend to cause fatal disease without good treatment options and are only handled in very specialized laboratories by specially trained scientists. You will not find BSL-3 or BSL-4 organisms in a college situation anywhere. BSL-4 organisms are only studied in labs equipped to handle them. BSL-4 labs are only in a few spots in the United States. This is one depicted here on this slide. You might notice that the scientists in this lab are in specialized suits that are pressurized. See how it looks like a balloon? There's air flowing into those suits and pushing outward. Why do you think that's the case? Why can't you just wear some tough, regular spaceship-like suits? The answer is, if you have air blowing into this suit, you are pushing air away from the person inside. Should there be any kind of small, even microscopic tear or rip, by pushing air away, you minimize the chance that anything could get into the suit and contaminate the person working inside. To work with BSL-4 organisms, it requires these specialized suits and a very specialized pressurized lab with filtered air to prevent organisms from escaping outside. Everything is highly controlled. These labs work with the most dangerous agents. Think something like Ebola. There's a couple other terms that have to do with heat and microbial control. Heat in general denatures proteins, but it also can make the membranes of a cell very loosey-goosey and fluid. Membranes that are too hot become leaky and lose their integrity. Heat also might disrupt the structure of nucleic acids by interfering with hydrogen bonding. When we're talking about heat, there's two terms to keep in mind. They're somewhat different, and you're going to just have to memorize these terms. First, the thermal death point is talking about one temperature point, a particular temperature, the lowest temperature that will kill all the cells in 10 minutes. Thermal death point is a particular temperature that kills all of the cells of interest to you in 10 minutes. The second term, the thermal death time, refers to a particular temperature and how long it takes to kill that microbe of interest at that temperature. For example, let's say you have a culture and you expose it to 60 degrees Celsius. You pick a particular temperature and then you determine how long it takes to kill everything in that volume at that temperature. That is thermal death time. Again, thermal death point is saying what's the lowest temperature that I can use to kill everything in 10 minutes. And the thermal death time is indicating that at a specific temperature, you need to remain at that temperature for a specific time to sterilize it. Please get these terms straight. As you can infer from the previous discussion, heat is a physical method of control. Moist heat is the first topic. Moist heat is more effective than dry heating at killing microbial cells. This is due to the fact that all those water molecules traveling as gas in the atmosphere are carrying energy that will conduct the heat, transfer it into the objects that the water comes in contact with. Moist heat with the water molecules is a better conductor of heat than dry heat. Boiling pretty much kills everything at 10 minutes if you're at sea level. Keep in mind that if you go up to the top of somewhere like Mammoth Mountain, then boiling will occur at a temperature lower than 100 degrees Celsius. Therefore, it will take longer to kill everything. At higher elevation, water boils at a lower temperature, therefore you need a longer time of boiling to kill all the cells compared to boiling at that same solution or boiling that same object at sea level. In boiling water, you can have survival of endospores and cysts because some endospores are very heat resistant and it's going to be difficult to kill them, even with boiling. The autoclave uses moist heat and pressure in order to kill. The autoclave is one of those instruments that is absolutely key to microbiology, so please understand this instrument. When you autoclave something, you're putting it in a chamber. This image is showing a very small apparatus. that might be used in like a doctor's office or a dental office. These would be used to sterilize something like instruments. There are some very large autoclaves in our laboratory at the college. Ours are capable of holding some very large flasks in order to sterilize a big batch of media for use in the laboratory classes. The idea is the same, whether the instrument is large or small. These are pressurized chambers that use superheated water. The steam will be at 121 degrees Celsius because you're able to get it hotter than boiling by keeping the steam under pressure. The chamber is held at 15 pounds per square inch for a minimum of 15 minutes. 121 degrees Celsius, 15 pounds per square inch, 15 minutes. That's a very typical autoclave cycle that should kill everything, including endospores. What you see down on this lower left of the slide is a set of controls to ensure that your autoclave is working. Here you have a vial that has liquid nutrient media in it. The vial also has a pH indicator in the medium, and outside of that inner vial, you have some endospores that are very heat resistant. To run a control of your autoclave, you would place this test vial into the autoclave when you run a cycle. When you're done, you remove that particular vial and take a fresh one that has not been autoclaved, and you break the little medium vial. You can do this by pinching it. What will then happen is all that medium will come out of that little vial and flood the endospore area. You then place both the vials in a high temperature incubator. If your autoclave cycle worked, then the endospores that went through the cycle are dead and nothing happens to the liquid media during incubation. The vial that did not go through the incubator should have growth of the endospores in the medium, which then changes color for easy reading. You can see on the bottom that this unchanged vial means that you killed all the spores because the color did not change in the medium. The one that did not go through the autoclave grew, indicating that the test vials are still working as you expected, and you can believe the test vial result. This is how we test our autoclave to verify their function each semester. It's time to end part 1 of chapter 9. We'll pick up with part 2 in the next mini lecture. See you soon.